Hello and welcome to Principle of Cognitive Neuroscience Part A. As you may know, this course is divided in 3 parts. First part, main models are all about understanding what people think about how the brain works, whether this is a brain that is separated in a mosaic of different regions contributing to different functions, whether those regions associate their activity in a serial way to achieve function as we know it, whether function emerge from the interaction between regions rather than their serial association, or are the neuron not really the core interest of function in the brain, but would it be rather the glial cells? And after all, what do we know about the functioning of the brain, and what do we don't know about the functioning of the brain? And those are like the five first lessons I give to you. Now we're on to the second part, which are the main concepts, which are mostly dedicated to what constitutes really a result in cognitive neuroscience that you will report or write a paper on. The previous course was about brain hierarchies, and now we're going to talk really about brain evolution. Every Monday we have practicals, and those practicals are live, and during those practicals really we're doing hands-on, and I teach you how to really do the research, and so those are not recorded unfortunately, but I'll invite you to connect and to join me. So brain evolution is a fascinating subject that I've been working a little bit on. This is fascinating because if you could travel through time as if each second were a million years, you know, our planet just appeared yesterday in that case, and then you're seeing things like rolling over you seconds after seconds. The first tree on the planet appeared 6 hours ago, and dinosaurs disappeared about an hour ago. Primates would have appeared less than a minute ago, with some only walking on 2 legs like less than 10 seconds ago. They did their first drawing 500 milliseconds ago and carved the first representation of a human face 250 milliseconds ago. That's a brass and Puy Venus that you can see here on the left. The relatively short span of our recorded history, everything that we know about us only happened in the 5 milliseconds. So we know nothing about the evolution of the human brain. And that's why it's so fascinating. Like everything has still to be done. Now, in terms of technical word, you might know the contrast between ontogeny and phylogeny. Ontogeny is a study of the development of the brain, of the development. Here we apply it to the brain. Phylogeny is a study of the evolution. And you have here like 3 main actors in the thinking of how brain or how evolution have been working, and then we can apply those principles to the brain. For instance, Lamarck and the Lamarckian thinking is pretty famous, since he thought that evolution relies on 2 different forces that will modify the body of the species. So for example, you take the giraffe, which is a typically strange animal because it's got a very long neck, and we can understand that this long neck is made to catch food in higher ground. And what was thought by Lamarck is the giraffe had a common ancestor, like in the old times, that was trying to stretch his neck to catch fruits and leaves, and this common ancestor transmitted this need to stretch his neck to his progeny, that transmitted the same need to his progeny, who transmitted the same need to his progeny, ended up with an animal with such a long neck. And so according to Lamarck, really evolution relies on two forces. One force, which is an adaptive force, whereby the body and the biology of the body adapt to the environment. And another force, which would be the elongation of the neck, and another force, which is a complexifying force, which with evolution, your body plan becomes more and more advanced and complex. And so this model of the evolution came into contrast with a different line of thinking, which is from Charles Darwin, who through the observation of species found a great deal of variability in the way the species could come in nature. So typically this is one of his drawings from his book, The Origin of Species, where you can see that the same butterfly comes up with like a different configuration of the drawing on his wings. And so his idea is like nature spreads this different configuration and select the one which is the most adapted to the environment. So it's a survival of the fittest. And this species that is the fittest to the environment will have the chance to transmit its inheritance and its characteristic through mating and having, you know, an heredity and, like, you know, kids, butterfly, whatever you want to call it, that will share the same specificity. And that's why you have white bear in the snow, in the ice ball, and this is why you have all these different species, and that's why you have a giraffe with a long neck. It's because the giraffes that had the longer neck were the fittest to the environment, had the better survival, were stronger, mated more, and then through natural selection and reproduction, this genetic difference was transmitted to the progeny. And so based on that, his idea was like, we can draw a tree, and that's what you see on the left part of this slide, where you have the common ancestor, which will be A, B, C, D, and so on and so forth at the bottom of the tree. And then you have, through evolution, different configurations that occurs and change, and like you have like a change of the biology of the species that occur, adapting to the environment, being selected, reproducing, transmitting its characteristics up to species of today. And then, like in the middle, you have a picture that I took in the Natural History Museum in Paris, where you have in the front a sea lion. I guess that's how it's called, notary, a sea lion. And you can see that the sea lion skeleton really much look like the hyena in the back. And those are like two species that live far apart in the different elements. The lion is more like the sea, the hyena is more the savannah, but they have the same similar skeleton because they have a common ancestor. Okay, different species, common ancestor, similarities. And this idea today is still used and thought about when looking at genes. Here you have one typical gene and you can follow its evolution. Well, you can see its variability on the right side. In human, you have different expression of this gene, and every color corresponds to a different expression of this gene. And you can find this gene in chimpanzee as well, and you have a match. The chimpanzee above that, the color, when it is the same color as for human, it is the same gene in human and in chimpanzees that you can find. And the same in the rhesus macaque on the top. The dark one represents unique genes that you can only find in chimpanzee or in rhesus macaque. And then by looking at this variability, you can see the one and how probably our common ancestors were looking like in 6 million years ago, and what like kind of genes existed 28 million years ago through the comparison of similarities and differences across genes. And we'll come back to this point a little later. And this was a revolutionary idea, and really Darwin was like a true genius and changed radically the way we look at evolution today. Without him, we'll be still thinking like Lamarck. And unfortunately, like any genius idea, at first there have been like some rejection from the public, and unfortunately Darwin during his living time have been suffering from a lot of ridicule of his ideas. You have here like a drawing from a journal called The Hornet, where they show Darwin as a monkey to ridicule really his theories that humans are coming from, the common ancestor of humans were like a monkey-like. And then another drawing that you can see here again during the living time of Darwin, from Charles Bennett, mocking again Darwin by showing this direct link and evolution between I think a goose and a human, and also like this parallel evolution between a trumpet and a body. And like, you know, it's just it is sad to see how great idea can be ridiculed, but it is also a lesson to learn for the future. If you truly revolutionary, if you truly provide new ideas and you change the way people think, you should expect some resistance and even unfair mockery from your peers, and you should not stop there. You should persist, because that means you're probably in the right direction. I'll tell you, when I started my PhD and started working on the white matter, people came to me and told me that I was an idiot. This will not go anywhere. And people will mock me and throw me tomatoes when I'm presenting in conferences. And look like where we are now. White matter is at the core of every analysis. I just think it's great that it turned around faster than it did for researchers of the 19th century. And then you got this third big man who kind of been able to build onto the Lamarckian and Charles Darwin concepts. Haeckel really proposed this vision that ontogeny summarizes phylogeny. So the way the biology of individual will evaluate is well, the way the body will develop from the infancy to the adulthood, from the embryo to the adulthood, is kind of like a fast version of how the evolution happened. So you have here like a page of the book of Haeckel, where you see like for the first time like a tree really representing evolution, and you see like different species with man really at the top of the tree. And in this next slide page of his book, you see like the theory that is pushing forward that we all start from an embryo, and this embryo, whatever the mammal species you are, is the same. So on the left you have a dog embryo, on the right you have a turtle, in the middle I think it's a duck, I'm not sure, and you can see they are like strictly identical. And what Haeckel proposed is that through the development you have this tiny change that will occur progressively to lead you from the same start of embryo to either a fish, a salamander, I don't know what are those, but I think it's a turtle and like a duck, and then you have a pig, like the rhino, I don't know what it is, but then you have a dog, and on the right side you have a human. And you see really like how interesting this development is. Again, his idea was ontogeny as a summary of phylogeny. He's the one who tackles this concept. However, you know, recently there's been some criticism onto his work. I just want to stress, if you look carefully, that this was published in 1875. It took like about 100 years to come back to this. And in this paper, the authors stress that Haeckel drawings were kind of modified to look like. For example, you say, well, real embryos do share many features at this stage, such as a tail and an identifiable body segment. They also have key differences. Human embryos, for example, have tiny protrusion called limb buds, particularly if they have developed to the point of having as many body segments as Haeckel gives them. But Haeckel did not include those limb buds, and in his drawing, the chick embryo is blackened like a mammal, but it wouldn't be pigmented that early. So, you know, like this author is claiming that there is a fraud, an exaggeration. A fraud would be a bit extreme, but there is a true exaggeration of the drawing from Haeckel, insisting on this relationship between ontogeny and phylogeny. Now, you know, like, how can we really study the evolution of the human brain? Because we can't really travel through time, can't we? And you know, apparently the development is not a good way to look at it. Yeah, you can try to measure it directly by collecting fossils of the skull of ancient humans. And you can see here this very impressive summary of the different skulls and periods that we find for the evolution of the human species. And you can see how you have different branches. It is not as linear as we might think. You have parallel branch, lost species, missing species inside this tree. And as you can see, each of those skeletons are dated to a different period, which allow us really to put them in a different place in the tree. For those of you who don't know, we can date bones and skulls thanks to a method which is looking at specific atoms and how they decayed, how much they decayed at the time the bone was formed. So if you don't know about this principle of physics, it's like, you know, like different atoms have different electrons, and some configuration of those atoms, like carbon-14, are a bit unstable, and they disappear and become carbon-13 quite fast, and they lose their extra electron, and then with time, their frequency in nature decreases. However, once they're bounded with the other atoms inside bones, they do not change anymore. So by doing a contrast between today concentration of carbon-14 and the concentration of carbon-14 in the sample of bones that we collected, we're able to estimate when these bones have been built or created in nature. So you have here like this nice charts that are showing you how much your carbon-14 existence in nature decreases. And so so if you know, we'll start from 100%. 5,700 years later, 50% of all the carbon-14 in nature disappeared, and then and so on and so forth until 30,000 years, when you only have 3% of the carbon-14 existing in nature. And that's what we call like half-life. It's how much time does it take in nature for your element to disappear. And here for carbon-14, it is 5,700 years. That's great. I really find that fascinating. But if you look carefully at these different skulls and bones, we're talking more about million years. And so like carbon-14 is no fit to quantify how old these skulls and bones are. So the way people investigate that, they use different isotopes. Those are like atoms with like with extra electrons. And for instance, they look at potassium 40, who has 1.3 billion years of half-life, which gives you like a better estimate of how long ago these skulls have been existing. And if you want to go even further, you can look at uranium. Uranium has the advantage, so that's your uranium 234, that has the advantage of having a half-life of 4.5 billion years. And so by using those two, you can estimate roughly when this skull was made and when like the individual you're collecting the skull from was alive. And that gives you all this information from which you can derive those trees and relationship. And then the relationship between the different species is based on the observation of similarities and differences in the bone structures, together, I guess, with also the geography. Where did you find these species? And that would be like studying the bones, but that still doesn't give us any information about the structure of the brain. We're going to have like maybe the overall size of the skull, but you know, where is really the difference? And to do so, you can infer from the shape of the skull what will be the shape of the brain. But contrarily to, on the contrary to what traditional phrenology has been indicating us, that like this is the outside shape of the skull that is giving us indication of that the shape of the brain inside the skull. You have like here another drawing that I showed you in the past lesson. Inside the skull, the brain leaves an imprint, which is called the endocast. So, as you may know, and we discussed that previously, the shape of the brain doesn't leave any trace on the surface of the skull, but, and this is really important, it leaves an imprint on the inside of the skull. So when you get those fossils, you can use them to do a cast. You know, people used to use like really wax and make like a cast of how the brain should have looked like. And here you have a digital version of it. So now we can do it with computers. And you can see how just with the skull, you can have a preview of the shape of the brain from a million years ago. And this has been done by Carson et al. It's been published about 10 years ago on different human species, particularly the Australopithecus sediba, where they were able, using really the skull, to reconstruct samples of how the brain looked like a few million years ago. Now, you know, you can look at the Australopithecus, which is probably one of the oldest humans, but you can also look at a more recent one, which is Neanderthal. And if you look at Neanderthal and you compare it with the brain of today, which will be the blue one on the right, you can see how the brain of Neanderthal was different in shape compared to the brain of humans of today. And you can even model it. There's been a paper from last year in PNS, and where you can see in green the regions that are mostly different in Neanderthal compared to the human Homo sapiens, which is us today. What's interesting is Neanderthal might be extinct, but during their living times, they've been mating with our species, and we all have a little bit of Neanderthal DNA that is coming from these ancestors. So I don't know if you know 23andMe, but so I did it. And so I decoded my genome, and I know that I have less than 2% of my DNA, which is Neanderthal, which is 211 variants in my DNA that trace back to Neanderthal. And this is important because this difference, these genes that are coming from Neanderthal define, you know, really the blueprint of my biology. So what really researchers have been trying to do in the papers that I just showed to you before is to see how Neanderthal allele in the chromosome 1 and 18, which are usually associated with reduced endocranial globularity, which is a shape really of the brain. Those two alleles, you know, they have some origin from Neanderthal, and you can really look at whether people that have those alleles have brain that look more like the shape of the brain of Neanderthal. And this is the case, as you can see here on the globularity score. You have a typical Neanderthal from which a brain has been reconstructed through the endocast, and we get this shape. And you can see that the people that have a brain that is closer to these cases, they happen to be more likely to have the genes, those genes of Neanderthal for the globularity of their brain. So it's fascinating how those two can be linked. And so future research can actually investigate really the relationship between the phylogenetic age, the estimated phylogenetic age of genes in our genetical code, and try to project that back onto the brain to identify and date and estimate the date of the different structures that we have in the brain of today. And there'll be the ultimate map of brain evolution as we know it. Let's get back to Darwin. So, because he said, we have common ancestors and species separate because, they take a different path of evolution, change the environment, different natural selection, and they don't crossbreed anymore because they're far apart, and then a new species appear at one point. It is possible to not only build the tree of the human species, but also build the tree of like all the primate species and look how they relate together. So here you have like all the different primates, some of the most important primates, and how, in time, when did they separate and came across like creating like two different species. And so like the idea would be essentially, if you were to compare pan troglodyte with Homo sapiens, the things that will be similar across those two species will be things that our common ancestors that we can see here in green had. And if you were to extend this comparison to like the comparison between Homo sapiens and gorilla, you will have similarities that belong to another common ancestor that was there a long time before that. So really essentially the idea is to surf along this tree to reconstruct and imagine really what will be the brain evolution, and travel through time and see this evolution happening, or imagining really this evolution happening. And so to do that, you need like a lot of species, and some amazing researchers have been investigating this point. And you can see in A that the work of Katia and Roberto Toro, and what they've been doing, they've been studying reconstructing the brain of different species of primates. They did this wonderful illustration on the left where you can see that different primates have different brains. They come in a different shape and size. They have common features such as a central sulcus that you can easily identify in all these brains, but the amount of circumvolution change. But mostly the first thing that is striking is the difference in size. Now, you can't make direct inference of the size of the brain itself. You have to do a ratio with the size of the body. And as you can see here in B, those primates come in different size. You have the bonobo that is between 40 and 60 kilograms, and the gorilla, which would be between 70 and 190 kilograms. And humans being between 50 and 90 at the adult age. So in order to identify really evolutionary trends, you need to do a ratio, and that will be in C on this slide, between the brain size or its weight and the body size or its weight here. And if you do a ratio, you'll see how all those species land onto this nice regression line. All the primates land like, maybe like a little above with mammals, and like fish and reptiles are on the lower part, but there is this very nice regression lines. And if you look carefully, and I circle it in red, the human brain is a little eccentric in the ratio between brain size and human and body size. So from this chart, you can extract two informations. There's been definitely an evolutionary change between fish, reptiles, and bird, mammals, and primates. There's been like something changed because all the white dots are in the lower part of the line, all the dark dots are on the upper part of the line. And that indicates a radical, fundamental change in the genes that define the importance of the brain inside the biology of the species, and here leading to bigger brains. And then the fact that humans are a little eccentric compared to other primates indicates that humans actually also benefited from a genetical change that led their brain to be disproportionate compared to the rest of their body. And then, like, so the question was really, where does it come from? Which part of the brain is disproportionate? Is that the entire brain? Is it a portion of the brain? And people put a lot of emphasis onto the study of the frontal volume, because the frontal lobe was thought to be really the area that is dedicated to all the abstraction, decision-making, control of the behavior that will theoretically characterize what it is to be a human compared to what it is to be a gorilla or a chimpanzee. And there's the same idea. If you want to measure that, you cannot just measure the frontal lobe and be like, it's bigger in humans. You need to do a ratio between the frontal volume and the overall brain volume. And if you do so, you are able to come up with interpretation of differences that happen across the species. And if you do that, you actually reveal that like the frontal volume is a lot disproportionate in humans compared to rhesus monkey and chimpanzees, although it is kind of discussed still. These differences between human and non-human primates can also be used to calculate maps of how much should you expand a rhesus monkey brain to match a human brain. And this work has been done by Vanison. It is and his team. It is in the line A, where you can see that if you wanted to take like a rhesus monkey brain and stretch it to make it a human brain, areas that I showed in hot onto this human brain here are the areas that you'll have to stretch the most in order to show this equivalence between the two species. The idea will be that those are the areas that have been evolving the most since our common ancestor with rhesus monkeys. So the areas that you see here highlighted in the hot color in the line A are the latest areas that appeared in the human brain on the evolutionary tree. In B, you have the same idea applied to the brain development, where you're looking at what are the areas that will require to be stretched to bring a baby brain to an adult brain. And what Vanessa has been really pushing forward and with his team is that there is a similarity, a resemblance between the line B, which would be the ontogeny, and the line A, which would be the phylogeny of an estimate of the phylogeny of the primate human brain. Line C is showing you areas that are showing a correlation, and you can see the area where you can see a clear relationship between brain development and brain evolution in red to yellow, and in green you have the areas that are showing an anti-correlation, which is where they are like not related in the same direction. Now, this is interesting. We're mostly looking at the surface of the brain here, but what is happening in depth of the brain? You know, you're now expert in neuroanatomy and white matter, and you might know, if you look at the hierarchy lecture I gave just before that, the frontal lobe is having this hierarchy of regions that are interacting, complexifying progressively the way you have your relationship with the environment, and increasing every time the level of abstraction and integration of rules to make the decision of what would be an appropriate behavior. So really what the next slide will be about is there a difference that will be highlighted into the human brain in the frontal lobe between the white and the gray matter compared to other species, and this is what we can see here. Smythe and his group have been investigating the ratio between the left prefrontal gray matter volume and the left prefrontal white matter volume between humans, apes, and monkeys. And what they've really been able to demonstrate is that apes and monkeys are clearly landing onto this nice line of regression, and there is a nice proportion between the left gray matter volume and the left prefrontal gray matter volume and the left prefrontal white matter volume. But humans, again, eccentric when we're looking at this line and show an atypically developed level of white matter connection, so exchange in the frontal lobe. And this suggests either a stronger hierarchical interaction that happened in the frontal lobe, which would be an associative vision, or higher level of integration if you were into looking at from an holistic perspective. So the next step from there was to identify which regions, which white matter pathways in the frontal lobe will be different compared to other primates, non-human primates, in human versus non-human primates. And so for the monkey, we have a nice depiction of the anatomy thanks to decades of axonal tracing. You know, you inject the brain area with a tracer and you try to look at how these tracers will follow white matter pathways in the brain. I encourage you to watch the fantastic lecture from Rockland that we had during our conference on structural connectivity, which is also on the Clinical Neuroanatomy Seminars YouTube channel, where you will see really those tracings and how it works. You have here one slice, and you can see like the injection is leaving this clear white tracings that you can see there. And the question is, how can you make the same thing in humans? You cannot inject during its living time a human brain, wait for this person to die, and then slice its brain to study how white matter is organized. That will be completely unethical. No, instead we use tractography of diffusion weighted imaging. The way diffusion weighted imaging works, I'll give you an example here. I was already showed those slides, just a reminder. It's measuring the way diffusion water molecule diffuse inside the brain. So as an example, if you are in the cortex where the organization of the neurons is a little chaotic, not completely chaotic, but a little chaotic, water molecule will diffuse randomly. Because they diffuse randomly, the MRI scan will record an equivalent direction of diffusion for all these water molecules that it can represent with a model, which is a tensor as a sphere, because there is no preferential direction of the diffusion of water inside that tissue. In contrast, in the white matter, you have axons that are densely packed, hydrophobic with water, like diffusing inside and in between the axon, but not across the axons due to the membranes that prevent the water to diffuse. So your water molecule will diffuse randomly, but mostly in the direction of the axons that you can represent with a tensor model as an ellipse, which is elongated according to the main direction of diffusion of water molecule inside that tissue. And then the idea is to follow one by one those voxel and the estimation and reconstruct huge white matter connections, as you can see here, representation of how white matter connects the different parts of the brain. Those two methods are not strictly equivalent, but this is the best we have. So at one point you have nothing better than, you got to use what you have. You're not going to wait forever, 50 years to have the perfect method. You do progress step by step and you accept and acknowledge the limitations. Here, main limitations are in the frontal lobe of the monkey, axonal tracing. We don't have a full vision of all the connections because we could not inject all the frontal lobe in monkeys. And this is because otherwise you wouldn't see anything, or you will need so many monkeys to inject that, it will be, completely unethical. In humans, you can see the full frontal lobe, but the spatial resolution of your tractography is moderate. You don't see single axons. What you actually see are bundle of 500,000 of axons together that are going in the same direction. And basically what we did, we used the atlas and the drawing of the atlas, which is called Fiber Pathways of the Brain. I definitely recommend this resource if you want to look at axonal tracing, and also if you want to learn about white matter. It is a fantastic book. And we just cleaned it up to focus on given connections that we wanted to compare with humans. And that's because your axonal tracing is not going to give you only the arcuate fasciculus, it's going to give you the arcuate fasciculus and any other tracks that are connected to the area you injected. For the MRI, we had a very nice subject. It was me who spent hours inside the MRI scan being scanned in order to have the highest resolution possible of diffusion weight and imaging at that time to be able to have the best depiction of the white matter pathways that you can see here on the right. And the work was mainly an anatomical work where you used to reach enough interest like we did during the practical to extract given pathways. And like the idea was to compare the anatomy of a single human, me, with the anatomy of monkeys as they were depicted for the white matter in the past 50 years. So in the result, we found similarities. And similarities are good. Well, one, because they comfort us that our method is correct and we really see true white matter connections. But they're also very good because similarities between the human and the monkey brain neuronal connectivity may account for the preservation of specific functions across evolution. So if you take, for example, the superior longitudinal fasciculus, it is organized in 3 branches in the monkey, SLF1, 2, and 3. SLF1 in red, SLF2 in yellow, SLF3 in green. And in humans, you can find the same connections. They follow the same direction. They are going in the same place. It is a system that apparently has been very well preserved across great primate species. And this system has been reported to be involved in visuospatial processing, in sensory motor integration, in working memory, and in eye movement in both species. If you were to disconnect those tracts, you will systematically in both species end up with visual neglect, apraxia, working memory impairment, and optic ataxia, whether you're in a monkey case or you are where you like voluntarily disconnect those tracts, or you're in a human patient where a stroke or neurosurgery disconnected those tracts, you will have the same symptoms. So here, similarity in the white matter connections between the two species seems to support the same functions, and it is very probable that our common ancestor 30 million years ago had exactly the same system. Then you have the Enzymate Fasciculus, which is also very similar between monkeys and humans. Same idea, Enzymate Fasciculus is connecting the ventral and medial frontal lobe to the temporal pole and the amygdala in the monkey and in the human. And in both cases, monkey and humans, the Enzymate Fasciculus has been associated with emotional memories and the estimation of risks. And if you were to cut the Enzymate Fasciculus, whether you're doing it in monkeys or in humans, you'll end up with memory disorders and antisocial behaviors, such as Phineas Gage. If you're going to the medial section of the brain, you can see this huge connection, which is called the cingulum, and the cingulum connects the anterior, the whole singular gyrus, anterior with posterior singular gyrus, and then dive into the temporal lobe to spoon really the hippocampus with some projection to the amygdala. And then what does this connection does? Well, in the monkey and in the human, this connection support a strong network, which is probably the most famous functional network, which is called the default mode network, which is very important for behavior and also for memory and the representation, like the representation of your individual inside space and time. And mind wandering. Then you have extra connections that we discovered at that time that were described, were visible in the monkey axonal tracing, but never ever reported in the human brain, such as the frontal ascending tract, for which function remain enigmatic in both species. The same goes for connections between the posterior part of the ventral frontal cortex and the frontal pole. Here, the orbitopolar tract, which has been, which is also visible in axonal tracing in monkeys and with tractography in humans, but for which functions still remain enigmatic. Now, that's it for the similarities, but we also found some very important differences. Unlike differences, you know, they might be related to the fact that we just use a different method to investigate those two species. But limitations set apart, those differences may also account for new functions that were elaborated in humans and that have no equivalent in monkeys. And so if you take, for example, the arcade fasciculus, well, the arcade fasciculus in the rhesus monkey, if you look at it first, it is not really arcade. And if you try to look at the equivalent tract in the human brain, you'll find that you have like just this little red segment that is very much looking like the monkey one. But the arcuate is also composed of a lot more fibers that we characterize as being the arcuate that are way bigger and connect many other regions in the frontal and the temporal lobe, which both have been drastically increased through evolution to lead us to have our human brain. Then if you were to compare the localization of functional or architectonic fields, it is true that they've been moving a lot through evolution. And if you compare rhesus monkey with chimpanzee and Homo sapiens, you see how, and then like with this, you have a better vision of how things have been changing. You see how the brain have been becoming bigger and stretching those connections to make it more important and support specific functions that are essential to our functioning as a human. I want to stress, if you lost perspective now, that like the scale of those brain is not the same. As you can see here, 1 centimeter is not the same across those three brains. I just put it into proportion so you can have a look and make a comparison. Next, if you move to the inferior fronto-occipital fasciculus that is described in humans, there is no equivalent of this tract in the monkey brain. Instead, what you have something that they call the extreme capsules that connect the frontal lobe to the temporal lobe and the posterior part of the temporal lobe, typically called temporal parietal occipital. And I want to stress here that you need to understand, and I really thank Michael Petridis for showing me this and make me completely understand what was going on. If you look at the occipital lobe, this is on the right part of the brain in the monkey brain and in the human brain. You see in the monkey brain these fissures that is separating the occipital lobe in two. And this fissure here is actually the calcarine fissure that we can see on the lateral portion of the monkey brain. And we cannot see it in the human brain because with evolution, the occipital lobe has been sucked into the medial portion of the brain. And through this movement, really, of the occipital lobe towards the posterior medial portion of the brain, it's been dragging, this is hypothetical, it's been dragging the external capsule more posteriorly and making these huge tracts that we call the inferior fronto-occipital fasciculus. And that's how evolution reshaped the white matter organization in the human brain. Now, let's think a little more. And I'll never stress that enough. Reading and thinking is as essential, even like more essential than just collecting data and doing analysis. So Charles Darwin has been saying, if the way evolution works, you get like this viability, like nature roll dice, and you have random change that happen in the genetical code that will have an impact into the biology of the organism of a given species. If that's a lucky draw, it will be an advantage, and the species will be fitter to the environment, survive and dominate more, have a larger number of progeny to transmit this modification of their genetical code and end up into a new species. If that's a bad draw, it ends up with a species like a biological system that is no fit, either die right away or is like weaker and does not survive, does not reproduce, and then this is the end of this genetical option. And that's the way you balance evolution. But really, if that is true, we should find some constant behind viability and the chance we have to find inter-individual differences between species. This is really something that we try to look at, which was like so how is one human brain different from another human brain? And is it the same for a rhesus monkey brain compared to another rhesus monkey brain? So if you look at how much you got to deform a human brain to look like another human brain, or a monkey brain to look at another monkey brain, if you look in terms of number of millimeters, you got to stretch it. You end up finding that, of course, Homo sapiens is more variable than a macaque, just because the human brain is bigger. But if you calculate a proportional expansion, if you put everything into proportion, like we said before, then the macaque brain is as variable as the human brain. And that's very interesting because I mean, it is a constant there that exists in the way change can occur in the biology of the shape, mostly, of the brain. And this is true whether you look at the gray matter or the white matter of the human brain or the macaque brain. So where do these change happen? They do happen in regions that are similar to the region reported by David Van Essen. They happen, and they do happen in similar regions in the human brain and the macaque brain, suggesting that actually areas that are variable, areas that are in development, really, and living changes that are comparable in humans or across primates. These changes look very much like the ideas that we have on how really the brain has been evolving, at least one of its theory, which is a dual origin theory, which suggests that the brain has been evolving from two different seeds. One, which we call the hippocampocentric system, starting from the hippocampus and then progressing onto the cingulate gyrus and radiating onto the medial portion of the brain to meet the middle frontal gyrus, coming across and meeting the frontal gyrus on the lateral portion of the brain. That'll be the white onto this drawing. And another trend, which is the olfactocentric trend, that is starting from the piriform gyrus within the depth of the lateral sulcus and progressively evolving onto the lateral portion of the brain to meet with the middle frontal gyrus and intraparietal sulcus in the lateral portion of the brain. And so if we look at the distribution of the variability, it looks like at the level of the hippocampus or the piriform gyrus, this variability is very small and purple. As you can see, the purple in the medial and the lateral view. Medial will be hippocampocentric, lateral will be the olfactocentric. And then as you're getting away from this center, your level of variability is increasing. And that would suggest, and this is what I like to call the Croxon theory, that would suggest that the more variable our brain regions, the later they appeared on the evolutionary tree, and that evolution is building new level of complexity and testing new configurations into these regions. So we try to validate this theory by doing a direct correlation between our gray matter variability and the evolutionary expansion map from David Van Essen, and showing that actually for the frontal and the parietal, the temporal, and the limbic lobe, we have significant relationship, at least for the left hemisphere and partially for the right hemisphere, between the viability and the expansion, how much you got to stretch over this monkey to reach the size of a human brain. So that will suggest that by mapping inter-individual variability, we can actually assess we can actually directly assess, have a map, if you want, of the brain evolution region being the most variable, being the latest developed on the evolutionary tree. And if you extend this concept to the white matter, I am pleased to show you for the first time the map of the white matter variability, which will be a surrogate for the map of evolution of the white matter. And there you can see that actually U-shaped projections are like those tiny connections that connect closed gyres, highly variable, showing that these regions have been changing a lot along evolution. Those connections have been changing a lot along evolution. And that like deep white matter structures, such as the fornix or the projection system, the corticospinal tract, are actually quite inviolable in the brain. And so you have the same, you have like here a dorsal view of this viability map, where you can see that also the structure that are viable are very comparable between the human brain and the monkey brain, except for the occipital lobe, as I said, that has been stretched and modified a lot from macaque to humans. We were pleased to make the cover of cerebral cortex with this result. An interesting thing is also, and we will have a lecture next about brain lateralization. It's really to try to investigate whether this variability is different between the left and the right hemisphere. And if this variability is different between the left and the right hemisphere, infers that there will be differences in the evolutionary trend of the left compared to the right hemisphere. If you do so, if you compare your two hemispheres, you'll find that the axial gyrus, of course, is asymmetrically variable in the left compared to the right hemisphere, but so is the superior and the middle temporal gyrus. So it is more variable in the left compared to the right hemisphere in humans. And the paracingulate and the lingual gyrus are more variable in the right hemisphere compared to the left hemisphere in humans. Really this difference in the temporal lobe will be related to the acquisition of reading and language, while the difference in the right hemisphere might be related to advanced emotional and social development in the human brain. But it is important to stress that macaque brain are not ancient human brains. They have their own evolution going on. And part of this evolution, when we compared the left with the right hemisphere, we were very satisfied to see that there are also significant differences. And the supramarginal and the angular gyrus are actually more variable in the right hemisphere compared to the left hemisphere. And that might be related to eventually advanced spatial abilities in the macaque, as well as new social development that is due to the selected breeding because those are macaques that are mostly selected. They're not coming from the wild, they're selected to do experiments. And these areas are important for theory of mind, at least in humans. So there will be, through this selective breeding, these areas will be the one that will be the most interesting for making experiments for humans, for having macaques that are willing to do the experiment and being performant at doing it, and being kind and understanding with their masters. To finish with the study of evolution, there is another vision that I found like also as very fascinating. It is a recent way to really investigate brain evolution that is called like a Neuro Archaeology. So essentially what Michel et al. Have been doing, so they use old carving from the prehistoric age that they showed to humans of today and try to decode through the activation of today's humans what those carving were. So those engravings, sorry, I said carving, but those engravings are compared to objects, visual scene, ancient scripts and words, and we can see that actually this these drawing are very comparable to ancient scripts and objects. So they actually were symbolic representation and not accidental drawings. And I find this approach of like decoding ancient artifacts thanks to modern fMRI absolutely fascinating. So as a take-home message for the study of brain evolution, I showed you like the wealth of different approaches that have been developed to study brain evolution with magnetic resonance imaging. So we spoke about the endocast imaging, which is taking an ancient skull and try to reconstruct, thanks to neuroimaging, how the brain was looking like inside this skull. We spoke a bit about the genetic neuroimaging, which is linking ancient genes with brain areas and trying to find what are those areas to like kind of date them thanks to the age of the genes that define their shape. Viability neuroimaging, that I like to call the Croxon theory, whereby we're showing that really the more viable an area, the more likely it is to be recent on the evolutionary tree, and the less viable an area, the more ancient it will be. This is an incremental building of brain evolution. Once you got find the optimal configuration, it doesn't really change it, it builds on it. Then you have comparative neuroimaging. It is all this idea, as you can see on the right, of looking at different species and finding similarities and difference to infer what happened since our common ancestor, what was shared with our common ancestor and what changed since our common ancestor. And then like finally, I give you this example of prehistorical neuroimaging where you show pieces of art or objects have been done from prehistoric time and try to see their interpretation through the network of activation that happen in humans of today. Now, you know, it sounds like yeah, fundamental science with no application to real needs of our society. And if you think that, you might be wrong. In the sense that understanding the evolution between species, particularly the brain, is the best way to identify the right species to do a pharmacological trial and study how to cure disease or model disease is only possible if we really understand the similarity differences that we have between species and our common evolutionary lineage. That's it for the lecture on brain evolution, and I will record soon the lecture on brain plasticity. I strongly encourage you to watch the special issue symposium on structural connectivity that we organize. You can rewatch it onto the clinical neuroanatomy seminar YouTube channel or website. And then I invite you as well to come every Monday morning to assist to the Neuro Keynote from 9:30 to 10:30 a.m. Paris time to discuss about exciting science of the last week together. This is a very agreeable time. To finish, I'll do a quick reminder that the Principle of Cognitive Neuroscience Part A will be the subject matter of an evaluation if you're a student in Padova, and that this evaluation is an integration of really your attendance to the practical course, participation to the class, which is whether this is during like the practical course or by asking question online on the YouTube channel or on the forum. And I'd like you to ask you to do a little project, which is to write an essay on the brain hierarchy of your choice. This is not about writing an essay about a single paper. This is about doing a true research on a hierarchy of your choice to write a nice essay divided in 5 sections with a total of 1,500 to 2,500 words. These 5 sections involve like a general introduction. You gotta make this hierarchy exciting for me. And a good way to test whether this is exciting, you should pitch people about your subject who are not expert on the brain or not really interested on the brain, and see whether this interests them or not. If this is not interesting for them, you probably didn't pitch it the right way and your introduction will not be optimal. So you got to find a way to hook people into your story, and that's good scientific writing. Then you have a section on the anatomy of the hierarchy, as it is very important to start with neuroanatomy. We're doing from anatomy to function here. Define it, what are you talking about, and what is the anatomical relationship between those regions in order to really understand the hierarchical relationship. Then, whoops, sorry. Then you want to investigate whether this hierarchy is lateralized. If there is no report of the lateralization of this hierarchy, I'd be glad if you can suggest how to do it. Be smart, be creative, suggest great ideas, and that would be giving you the highest grade. And hierarchy is interesting in the way it links with evolution. It is a good model to study and to link with brain evolution. I would like to also see in your essay a section about that. How does this hierarchy is linked with brain evolution? Then you finish with a conclusion, a synthesis, where you bring everything together, make sense of the relationship between the anatomy, the lateralization, and the brain evolution together in the hierarchies, and you look at. And that will be great. If you have any question, you can send it to me to michel.thiebaut@gmail.com. And I'm expecting, of course, your essay before December the 11th of this year, submitted and sent to my personal address, michel.thiebaut@gmail.com, before midnight. With this, I thank you for your attention, and I'm looking forward to see you doing the practical or at the next lesson.